Okay, today we're working with cell structure and function. This first page of this assignment just gives you some general information. You'll need to read through that. The next page, if you'll notice, we have a bacterium, and there are two types of cells on the Earth, actually. The first one is the prokaryotic cell. That includes only the bacteria, and they're very simple, the simplest cell. And the second cell would be the eukaryotic cells, and that would include all the other cells, which of course would be the plant, the animal, the fungus, and protists, which we will get into those. So let's look at the prokaryotic cell, the bacterium. If you'll notice, there are several structures there, and they have those labeled, and they also have the functions of all those listed. There is only one DNA molecule. There is no nucleus on that cell whatsoever, so the DNA is not encapsulated with a membrane at all. And these are structures that you might find on a bacterium. Structures that all bacteria have in common would be the single DNA molecule, and that is a continuous loop. It looks like a bunch of threads in there, but that is only one, and it is a continuous loop, meaning it doesn't have free ends on it whatsoever. And then of course you have a plasma membrane, which would be this structure we're looking at right here, and then a cell wall. So the nucleus, the plasma membrane, the cell wall, those are structures that are common to all bacteria. Now, in addition to that, other bacteria may have other structures. For example, you see this layer here that's called a glycocalyx. That can either be very dense, which they call it a capsule, or it can be less dense, where they call it a slime layer. So depending on the bacterium, you have either capsule or the slime layer. You notice all these hair-like structures around the bacterium. Those are fimbriae. Those are used to adhere to structures. Say like possibly if you were to get a bacterium into your oral cavity and it had fimbriae, then it would actually adhere to your mucous membrane of your oral cavity, and then every time you would swallow, it would not go down with the mucus and the saliva. So it would stay on the mucous membrane for a long period of time, and then therefore it would be difficult to eliminate, and it wouldn't go down into the stomach, which is really a pit of acid, and be destroyed. This structure here would be a flagellum, which is very different than a eukaryotic flagellum, and we will discuss that in lecture. But keep in mind, this is the prokaryotic cell. There are a few other structures. You'll want to read through that and identify those. Okay, now when we get into the plant and animal, and they don't go through the fungus here, just plant and the animal, they're going to give you a list of cell structures, and then also the functions, composition, and then they give you the functions of these structures. Okay, if we turn the page, what you'll see is this is an animal cell, and they have labeled surrounding this A, B, C, D, and so forth. Okay, A would be the nuclear envelope, sometimes called the nuclear membrane. B is the nucleolus, that's where ribosomes are produced. And then C is going to be rough endoplasmic reticulum right here. This is where the proteins are manufactured and may be packaged for export. And then D, this is called a smooth endoplasmic reticulum. It's used for detoxification of poisons, and also production of phospholipids, and also any kind of a fatty compound. There are enzymes in there to produce those things. Okay, letter E, there's your plasma membrane. There is no cell wall on an animal cell. Letter F, that would be our mitochondrion, and that's where the ATP is produced for the cell. Letter G, that's called the Golgi with a capital G, Golgi apparatus, And that's kind of a repackaging center where they can package, say, sugar and fat, or sugar and protein, or protein and fat, a packaging center for that. And then they have budding off of these little flattened saccules, we have these little buds, and those are called vesicles. And those vesicles could be transported either out of the cell, which is what's happening here, or be used within the cell. And in addition to that, let's see, we have the centrioles. Those are used in cell division. And then the peroxisome, we'll actually detox the hydrogen peroxide, which is produced by cells. And in addition to that, you see all of these little fibers that run through this cell, and those are used to anchor organelles into a specific spot and maintain the shape of the cell. Not all animal cells are round. Some of them are square. Some of those are also going to be elongated. Some cells are flat, so something has to hold those into that three-dimensional position. So we call that the cytoskeleton. And it's, again, these old fibers that you see in here that crisscross throughout this cell. Okay, then on the plant cell, they have everything that the animal cell has, but in addition to that, you'll notice they have this big, huge sac within the cell. It's called a central vacuole, and that's a storage site, mostly for water. And then you'll see the green structures. These are chloroplasts. That's where the plant cell produces its sugar by photosynthesis. And then if you'll notice, there's a outer lining to this. You've got a plasma membrane, but exterior to that you see this, which would be the cell wall. In reality, it would not be green. I think they just did that to make it visible. But the cell wall maintains the shape of that cell, and not that they don't have the cytoskeleton, but it does help to maintain the shape of the cell. It's called cell wall. All right, now when you get over to the portion of the lab that is plant cell structure, there isn't anything that we need to change on that, except number 9 where it says save your slide. Don't worry about that. You won't have to be saving that slide. We can make other slides. Okay, the other thing you'll be doing is working with processes by which things can get in cells and out of cells. For example, this is just a diffusion, which is a movement of a substance from where you have a high concentration of a substance to where you have a low concentration. So what someone has done is put a dye in this, I would have guessed that would be a petri dish. And in the center of that, those are the red spherical structures, and then over time the dye begins to spread out into the areas where the concentration is low or where there isn't any of that dye, and eventually they're equally distributed, so eventually it does reach an equilibrium at some point. Okay, so you will be doing that with a semi-solid, which will be an agar plate. You'll put a depression in that, and I'll show you how to do that. Put a little bit of dye in that depression and see how that works. You'll use a liquid, which will be distilled water, and you'll put some dye in the center of that and measure diffusion rates. And then also we will diffuse through air. We'll use just an air freshener and see how fast that air freshener moves through air. Okay, now on this lab right here, this little portion of the lab, what you're doing is you're putting substances within a tube, which is called a dialysis membrane, and within that you're going to put, you can read it here, glucose and starch, and then outside you'll put water and iodine. So inside the bag you have glucose and starch, and outside you have water and iodine. So what we want to see is what substances can move into the bag and what substances can move out of the bag. And you'll do a little testing on that. Just follow the directions. Again, we don't need to alter those directions at all. And let's see. Okay. Osmosis is the diffusion of water. So water will move from an area of high concentration to an area of low concentration, pretty much like what the dye did, except that we're using water, of course, instead of dye. So from a high to a low concentration. This portion of the lab right here we're not going to do, so actually you can omit that section, so we're not doing that. On tonicity, if you'll notice the term isotonic, iso means same in Latin. Tonicity refers to the concentration of the solute, so whatever's dissolved in the water. So if I had, in this case, if I had a 0.9% salt dissolved in water, that is isotonic to red blood cell water concentration, because the concentration of red blood cells is a 0.9% salt. So you're not going to get any movement of water into or out of that cell. Alright, and they do want us to use red blood cells. We will not be doing that. We used to do that when we could prick our finger with a lance, but we're not doing that, not these days. Okay, experimental procedure, effective tonicity on red blood cells. Again, we are omitting that, so we're not going to work with that. And then we are going to work with the Elodea, which is a plant that's a water plant. So we will work with that. And then what we're doing is we're working with what is called a hypotonic. Now hypo means below, and tonicity again means concentration of solute. So that means you have a concentration of solute that is less than what it is in the cell. So for example, if you put a pure water on top of Elodea, and then put the Elodea on the microscope with a drop of pure water on it, well, pure water has nothing dissolved in it, so it certainly has less solute than does the cell. So you'll be able to notice what happens with that. Okay, and then in a hypertonic solution, hyper means more, tonicity means concentration of solute. So obviously you have more solute in the solution. So if we have, let's say for example, a 1% solute concentration within the Elodea cells, and then that gives us a 99% water concentration. If we put a 10% salt concentration on it, that obviously is going to give us a 90% water concentration. And let me show you how that actually works on paper here. So let's say that this is the Elodea cell here, and we'll put in a 1%, we'll say solute. That can be sugar, it could be amino acids, it could be salts, but anything that dissolves in the water. So I have a 1% solute, 99% water, and let's say outside the cell I put in a 10% solute, say salt, it could be sugar, it could be again amino acids, something that dissolves in the water, and let's say a 90% water concentration. Then what happens, water is going to move from where you have more of it to where you have less. That's called osmosis. So the water in this case is going to go out from inside the cell to outside the cell, so it's going to move out. So if I put a plant cell like Elodea into a 10% salt solution, which of course is 90% water, the 99% water that you have inside the cell will move out of the cell, and that's just osmosis. Water moves from where you have more of it, again the 99, to where you have less of it, which would be the 90. And what that does is shrivel up the cell. That term is called a plasmolysis. Plasmolysis. This is what you would call a misnomer. It's not a good term because lysis means to break, and plasmo means you're breaking the plasma membrane. You don't actually break the plasma membrane, it just shrivels up is what happens. So the plasma membrane would just shrivel up very close around this because you will have lost a bunch of water, kind of like letting water out of a water balloon. So the entire plasma membrane would be pulling to the interior. Okay, now let's look at the other situation where we had a hypotonic solution. So in a hypotonic solution, again in a Eladia, we're going to have, say, like a 1%. That would be solute, so that would be sugar and salt and amino acids and whatever's dissolved inside that plant cell. Again, that's a 99%. Water solution, that's pretty common for cells. And then outside, we're going to place 100% pure water. So this is distilled water. This has nothing dissolved in it. So that means that what we have is a 0% solute, which means nothing is dissolved in the water. It's just pure water. Now, osmosis tells us that water is going to move from where you have more water to where we have less water. So in this case, the water is going to move in, and it creates a pressure inside of this, and that pressure for a plant cell is called turgor, Turgor, turgor pressure. So the water moves in, creating the pressure, and this is in a plant cell, and that's called turgor pressure. Okay, well, let's look at the same situation, except let's work with an animal cell instead of a plant cell, and pretty much have the same event occur. So let's do that again, except we'll use the same concentrations to keep it simple, but this is an animal cell here instead of a plant cell. So again, cells typically are about a 1% solute, and that is a 99% water. Okay, and then outside we're going to place a 10% salt. So that's my solute. I'll just put solute. Could be sugar, 10% sugar. That's a 90% water. And remember, osmosis, again, is the diffusion of water. So water moves from where you have more of it to where you have less of it. Always from a high concentration of water to a low concentration of water. So it's going to move out. Of course, what this causes is the cell just to shrivel up. Well, in in animal cells, we call this a crenation. Again, that is a shriveling, shrinkage, however you'd like to describe that. So this plasma membrane, there is no cell wall. The plasma membrane just pulls to the interior since you are losing water. Again, it's like letting water out of a water balloon. Okay, so let's look at the other situation where we have an animal cell. Again, the interior is approximately 1% solute. Inside the cell, we have a 99% water. All right, and then outside, we're going to place 100% water, distilled water, has nothing in it, no sugar, no salt, nothing there. Okay, so that's a 0% solute, nothing dissolved. Okay, so water is going to move from where we have more water to where we have less water. So water moves to the interior of the cell. Well, animal cells do not have a cell wall on them, so if we bring water into this cell, it's going to fill up with water, and then eventually it will break. It's like putting too much water in a water balloon. It does burst. Now, in the case of the animal cell, we don't call that a plasmolysis. We call that just a lysis. It just breaks. It breaks. To lyse, to break. Okay, so that's what's happening with that. Okay, now back to the lab. You do want to do the experiment using the potato strips. That works very well as written. Nothing to change on that. And then at that point, I think given the time frame that we have in order to do this lab, I think that will be the last part of this lab that we will do once you get to the potato strips. Okay, thank you.